Ladies and gentlemen, please give a warm welcome to the laureates and laureate representative for the 2018 Tang Prize in Biopharmaceutical Science, Dr. Tony Hunter, Dr. Brian Drucker, and Dr. Ming-Chi Hong. And please welcome our host, Professor Xu Qian. Good morning, distinguished guests, ladies and gentlemen. Welcome to join us at the Laureate Lecture of the 2018 Tang Prize in Biopharmaceutical Science. To begin with, we would like to invite to the stage the host of this lecture, Dr. Xu Qian, University Professor of the University of California, to give us the introduction. Ladies and gentlemen, please give a warm welcome to Professor Qian.
Good morning, ladies and gentlemen. It's indeed my great pleasure and privilege to host this 2018 Tang Prize for Biopharmaceutical Science Laureate Lecture this morning. The 2018 Tang Prize laureates in biopharmaceutical science. Laureates are Drs. Tony Hunter, Brian Drucker, and John Mendelsohn, for the discovery of protein tyrosine phosphorylation and tyrosine kinases as oncogenes, leading to successful targeted cancer therapies. Dr. Tony Hunter made the groundbreaking discovery of protein tyrosine phosphorylation and tyrosine kinases in the late 1970s. This sowed the seed for research in the ensuing 40 years, leading to a thorough understanding of the fundamental principles of cell growth and cancer development, as well as the notion that blockade of tyrosine phosphorylation can be a therapeutic approach for cancer. Dr. Brian Drucker made important contributions in the use of tyrosine kinase inhibitors, TKI, to block tyrosine phosphorylation as a targeted therapy. He led the successful clinical trial of the antibody imatinib, or Gleevec, which inhibits ABL tyrosine kinase on chronic myelogenous leukemia. This is a prototype that opened up the field of TKI as targeted therapies, which already have significant impacts on the treatment of cancer patients. For tyrosine kinase to function, it needs to bind to the receptor tyrosine kinase, RTK. The development of antibodies against the extracellular domain of the RTK to prevent the binding of the oncogenic growth factors would be another way to block the tyrosine kinase activation. Dr. John Mendelsohn used antibodies targeting epidermal growth factor receptor, EGFR, which is often overexpressed or mutated to become oncogenic. He conducted preclinical research and developed the anti-EGFR antibody cetuximab, which was approved by FDA in the United States for the treatment of colon and head neck cancer, as the first clinically approved therapy based on RTK targeting antibodies that spurred many others. The development of tyrosine kinase targeted therapies has fundamentally changed the practice of cancer therapy. It provides great benefits to patients who suffer from this dreadful disease and gives hope that cancer can eventually be treated. The superb accomplishments of the three laureates of the 2018 Tang Prize in Pharmaceutical Science have made immeasurable contributions to science and society. The achievements of the three laureates clearly illustrate how brilliant basic science can lead to effective clinical applications that benefit all mankind. we have the great privilege today to hear the 3 Laureate lectures from Dr. Hunter, Dr. Drucker, and Dr. Hong, who is going to speak for Dr. Mendelsohn. So I will first make a brief introduction for Dr. Tony Hunter. Dr. Hunter was born in the United Kingdom, and now he is a citizen of the United States. He received his BA and PhD from the University of Cambridge and was a research fellow there in the Department of Biochemistry. In 1971, he came to the Salk Institute in San Diego of the United States to become a postdoctoral fellow. Then he became a faculty in molecular and cell biology, becoming a full professor and Renato Tobacco Chair in cancer research. In 2008 to 2016, he was the director of the Salk Institute Cancer Center. And he is also an adjunct professor in 2 departments in the UCSD in San Diego, in biology and pharmacology. He has received many awards and recognitions, including the Gaitner Foundation International Award, the American Cancer Society Medal of Honor, the Kirk Langdon AACR Prize for Basic Cancer Research, the Louisa Gross Horowitz Prize, the Passero Award for Cancer Research, Clifford Prize for Cancer Research, the Royal Medal in Biological Sciences, the Schussberg Prize for Cancer Research, and the most recently, the Pezzocular AACR International Award for Extraordinary Achievement in Cancer Research. He is a Fellow of the Royal Society of London, the American Academy of Arts and Sciences, and National Academy of Medicine, and National Academy, the American Philosophical Society, as well as the AACR Academy. As mentioned, he gave birth to the field by discovering tyrosine phosphorylation and showing that the oncogene SRC is a tyrosine kinase. So he has really led a field that allow us to be able to do all the things in the treatment of cancer using this important basic discovery he made. His talk today is entitled "The Tyrosine Phosphorylation from Discovery to Drug Development and Beyond." Let's welcome Dr. Tony Hunter.
Good morning, everyone. Let me start by saying what an honor it is to have been selected as one of the 2018 winners of the Tang Prize for Biopharmaceutical Science. I'll start by giving you a brief history of the discovery of tyrosine phosphorylation and then talk about some of our own ongoing work. So the story begins in 1979. I was at the Salk Institute as an assistant professor, and we were working on polyomavirus, a small DNA tumor virus which encodes 6 proteins, with the hope that we would learn something about the basis of human cancer. And we and others had shown that one of the 3 proteins made from the so-called early region, middle T antigen or middle tumor antigen, was responsible for the transforming activity of this virus in cells in culture. And this is what I look like, in case anyone's wondering. I grew my beard at the bottom of the Grand Canyon in 1972. The last day I shaved was June the 21st, 1972. So we had a protein that we knew could cause cancer, but we didn't know how it worked. A year previously, Mark Collette and Ray Erickson had shown that the transforming protein of a second tumor virus, Rouse sarcoma virus, called VSARC, had an associated protein kinase activity. And this was an exciting idea that suggested maybe other viruses use protein kinases to transform cells by altering protein activity. And so we tested whether the polyoma middle T had such an activity by isolating it in an immunoprecipitate and incubating it with gamma p32 labeled ATP. And in this lane C, you can see that the BAM became heavily p32 labeled. We had mutants of polyomavirus that do not transform, and these mutants here lacked the kinase activity, correlating it with the transforming activity. So we weren't the only people who had this idea, and two other groups, Alan Smith and Mike Fried and Brian Schaffhausen and Tom Benjamin, had also tested this idea and had found that the middle T protein had an associated kinase activity. That summer, there was a meeting at Cold Spring Harbor on tumor viruses, and we all got together, and after giving our talks, decided we would co-submit our papers to Cell when we got back, and we submitted ours on June the 11th, 1979. So we got back the reviews, and this is what they said. These two findings force one to seriously consider the possibility that this is an in vitro artifact. The product of this in vitro protein kinase-like reaction was not characterized, and with the very low activity, it might be the result of some contaminant. And the third review was, unfortunately, very few of these conclusions drawn by the authors are actually clearly substantiated by the data. So you would have thought the paper would now be rejected out of hand, never to see the light of day. But the editor of Cell at the time was Ben Lewin, and he was an autocrat, if you like, and he decided he liked these papers and wanted them to be published. So one of the criticisms was that we hadn't characterized the activity, and I already knew that this was a likely criticism, and so the day after, I seem to be now, yeah, so you can't read this, I know, but the day after we submitted, on the 12th of June, 1979, I set out to find which amino acid was phosphorylated by taking some of the labeled band, hydrolyzing it in acid, and separating it on a thin layer plate by electrophoresis at pH 1.9. And if you could read it, that's what this says. So the plate, after separation, was exposed to an X-ray film together with stained markers for phosphoserine and phosphothreonine, the only two known phosphoamino acids, and this was the result of an overnight exposure. Now, you might look at this and see nothing, but in fact, with the eye of faith, there is a weak spot here. And with a longer exposure, you can actually see that this spot migrates between phosphoserine and phosphothreonine. So this suggested there was some new product of this kinase reaction that was acid stable. And so I said something like, you know, clearly this is not phosphoserine or phosphothreonine. So it could have been an artifact, but I repeated the experiment a few days later and got the same result. So since this was stable to acid hydrolysis, it probably was a phosphate ester linkage. And the most logical possibility was tyrosine, which was the only other hydroxyamino acid that I had learned in my days in the biochemistry department. So serine and threonine are phosphorylated on the beta hydroxyl group. Tyrosine has a phenolic hydroxyl group, so it's very different in one sense. So to test whether this new spot was phosphotyrosine, I tried to make some phosphotyrosine by incubating tyrosine with phosphorus oxychloride, and discovered this gives you a black tar. I'm no chemist, as you can realize, but in the end, I did purify a little phosphotyrosine, and in fact, the spot co-migrated with the radioactivity. So, on the verge of something important, I decided to go and raft a river in Idaho. And on July the 3rd, a group of us drove up to the Salmon River in Idaho. And here I am, rowing my boat down the Salmon River in Idaho. Didn't get back for a whole month, in fact. when we got back, I realized I needed a better separation for phosphotyrosine and established this two-dimensional system, which clearly shows that the radioactivity from middle T co-migrates with tyrosine and not phosphoserine or phosphothreonine. So we resubmitted the paper to Cell with these new data in it on September the 25th. It was accepted on September the 27th, which means that it was not re-reviewed, and this was Ben Lewin's decision. The other two papers from the other groups were also resubmitted, but they did not, or had not been asked to identify the phosphoamino acid. And so these papers were all published in December of 1979. So in retrospect, the reason why I had discovered phosphotyrosine was that I had been too lazy to make up a fresh buffer. And the pH 1.9 buffer I had used many times before changes pH and drops to pH 1.7. And this allows phosphotyrosine to resolve from phosphothreonine. Turns out the activity of middle T is not intrinsic, but it's due to an associated protein kinase that Sarah Courtney showed was the product of the CSARC gene. So in the course of doing those studies, I realized that I ought to try another method of isolating phosphotyrosine from the labeled middle T, and carried out a complete protease treatment to release amino acids, and ran that sample out, and you can see the band or the spot co-migrates with phosphotyrosine. But at the same time, we were working on the Ras sarcomavirus VSARC protein kinase, and so I thought I'd just include that as a control. In this case, the VSARC protein phosphorylates the heavy chain of the precipitating antibody. And when I ran out that hydrolysis, much to my amazement, it turned out that this was phosphotyrosine and not phosphothreonine, as had been reported by Erickson and Collette. So that was done on September the 18th, 1979. So this autoradiogram and the thin layer plate from which it was derived are now on exhibit as part of the Tang Prize show in the Chiang Kai-shek Memorial Hall. So from this, it looked like SARC was also going to be a tyrosine kinase. And here is additional evidence that this is phosphorylating tyrosine. And more importantly, we were able to show that chick cells transformed by the VSARC protein had elevated levels of phosphotyrosine in cellular proteins based on this two-dimensional separation, showing a small amount of phosphotyrosine in the uninfected chick cells and about a 10-fold increase in the transformed chick cells. So we submitted a paper describing this tyrosine kinase activity of VSARC to PNAS, and these reviews were a lot kinder. These findings are both interesting and potentially extremely significant in that phosphorylation of tyrosine has never been reported. The primary significance of this work derives from the fact that the phosphorylation of tyrosine is such a novel activity. And finally, a handwritten review saying no revisions necessary, publish as is. So this paper was communicated to PNAS by Bob Holly and appeared in March of 1980. So amazingly, all the experiments, starting with the first experiment on the polyomavirus kinase activity to the studies of the VSARC protein, took less than 5 months, and all of the experiments in the VSARC tyrosine kinase paper were done in less than a month. If anyone's interested in the history, here's a review I wrote a couple of years ago. So by now, I was really interested in protein kinases. And so at about the same time, the first sequences, both protein sequences and DNA sequences of proteins with protein kinase activity began to appear, starting with the sequence of the cyclic AMP-dependent protein kinase down here. And here's the sequence of the VSARC tyrosine kinase. And I began to align these sequences to see if they were related to one another. This was done by eye, so BB here stands for before blast. And it turns out that the human eye is very good at aligning sequences, and you could see all of these kinase sequences had these related motifs along the length of the catalytic domain, and it turns out these motifs are conserved in all of the eukaryotic protein kinases and are used as a hallmark of a protein kinase when a new sequence is obtained. The first report of these sequence alignments didn't appear in a regular journal, but instead appeared in the Scientific American, where I wrote an article on the proteins of oncogenes. So by using these sequence motifs, these catalytic motifs, it began to be clear there will be a very large number of protein kinase genes in a single genome based on the rate of appearance of new reported kinases. And so at the time, I somewhat rashly predicted there might be as many as 1,001 protein kinases. When the human genome was completely sequenced in 2002, we could actually deduce how many protein kinases there are, and it turned out there were 478 protein kinase genes in the human genome, closely related to that original alignment I showed you, as well as a few atypical protein kinases, many very distantly related, including the PI kinase-like kinases, ATM, ATR, mTOR, and DNA PK. Interestingly, about 10% of these kinases are predicted to lack catalytic activity because they're missing one or more of the motifs. And these have become known as pseudokinases. Many of these have important scaffolding functions. Fast forward another 16 years, and we, and particularly Pryor's group, have been able to identify some additional protein kinases in the human genome, and the number stands now around 535, not quite 1,001, but not so far off. All of the new protein kinases are remote or atypical protein kinases, and I'll briefly tell you about some work we've been doing on the enemy family of histidine kinases. So how many tyrosine kinases are there? So we had one in 1979. By the end of 1980, there were 4, including 3 new viral tyrosine kinases and the EGF receptor that John Mendelsohn worked on. And when the complete human genome sequence was reported, it turned out there are 90 tyrosine kinases, and you can see them here in the tyrosine kinase branch of the tree. Over half of those are implicated in disease, and particularly in cancer. And it became clear from surveying the literature that mutations in protein kinases would be causal in a fairly large number of diseases, and particularly in cancer. And this led to a lot of interest by pharma and academia, actually, in developing small molecule inhibitors of particularly tyrosine kinases as possible therapeutics. And as of a month or so ago, there are now 45 approved kinase inhibitors, 32 of which are TKIs or tyrosine kinase inhibitors. And I don't suppose you can see this, but starting over here with imatinib or Gleevec that Brian will describe to you, you can see there's just been a steady increase in the number of kinase inhibitors, and there are many, many more in clinical trials. So imatinib was the first targeted cancer therapeutic, and now we have a whole collection of kinase inhibitors that target other kinases and other cancers. So with that introduction, let me now turn to the 2 ongoing research projects in the lab. First one is a more translational project to try and develop better therapeutic treatments for pancreatic cancer, one of the most devastating cancers, largely because it's usually diagnosed so late. Pancreatic cancer shows a progression of phenotypes and morphologies and characteristic genetic changes, including a KRAS activating mutation, loss of a CDK inhibitor, and P53 and SMAD for loss of function mutations. And the disease goes through these morphological features called pannins. Ultimately, the cancer, which is in situ, becomes invasive and begins to migrate through adjacent tissue and then potentially into the bloodstream to establish metastases, usually in the liver. So I should remind you that solid tumors don't just consist of tumor cells. They also contain stromal cells of various sorts, including blood vessels, immune cells, and cancer-associated fibroblasts, and particularly in this case, a specialized stromal cell called the stellate cell. Stellate cells are resident cells in the pancreatic tissue. They have a star-like shape, hence their name. These cells are activated in response to inflammation or the formation of a tumor. They are highly secretory cells, and they pump out extracellular matrix proteins as well as a large number of cytokines. And they are responsible for forming a dense stromal matrix around nests of tumor cells in an attempt to wall off the tumor cells. So Yushi, a postdoc in the lab, became interested in whether proteins secreted by the activated stellate cells, particularly cytokines, could act as a stimulus to the tumor cells to promote their proliferation and survival. And we thought that paracrine factors of this sort might make good targets. And I'm going to focus on a protein secreted by the activated stellate cells called leukemia inhibitory factor. It's a cytokine in the IL-6 family. So our initial approach was to carry out an analysis of all the secreted proteins from both tumor cells and a stellate cell line using mass spec. These secretomes, as we call them, are very complicated, with over 2,000 proteins secreted by the tumor cells and 1,400 by the stellate cells. And we focused on proteins that were unique either to the tumor cells or to the stellate cells. And you get a long list, and you have to choose something to prioritize. And so we chose leukemia inhibitory factor because it appeared to be unique to the stellate cells and has interesting stem cell properties. And just note, in addition, that the stellate cells make a lot of extracellular matrix proteins, as we already had expected. So why do we focus on LIF? We knew that pancreatic stellate cell conditioned medium can stimulate the phosphorylation of STAT3, which is a LIF target. And we could show that antibodies to LIF neutralize this activity. We identified the LIF receptor as being bound to a protein in the stellate cell conditioned medium, and we showed that the stellate cell secretome has high LIF levels. And I'll just show you that here, indeed, by an ELISA assay, the stellate cells have a lot of LIF. We had used MIA-PACA tumor cells. They have very low levels of LIF, but you can see there are tumor cells that also make LIF, and so LIF is very high in pancreatic tissue, as I'll show you, and it may be produced by both the stromal cells and the tumor cells. So LIF, or leukemia inhibitory factor, is a stem cell factor. Some of you in the audience may use LIF to maintain the pluripotency of ES cells grown in culture. And so we thought it was interesting from that perspective. Perhaps it was activating stem cell-like cells in some of the tumor cells. As I said, LIF is a member of the IL-6 family. They all use a common signaling subunit called GP130, and then they have a specialized ligand-binding subunit. And so each of these receptors, while signaling through the same subunit, all bind to distinct heterodimers. And the major signaling pathway activated by LIF and IL-6 is the JAK-STAT pathway. Coincidentally, Jackie is a tyrosine kinase and phosphorylates a tyrosine in the STAT transcription factor, leading to its dimerization, migration into the nucleus, where it activates expression of stem cell genes like SOX2 and OCT4. So having shown that there's a high level of LIF expressed in pancreatic tumor tissue, we wanted to know whether this is actually important as a driver of pancreatic cancer. And so we used the mouse model developed by David Tuveson called the KPC model, in which PDX1 Cre, which is expressed during the development of the pancreas, activates expression of a G12D KRAS mutant gene, and at the same time deletes both copies of the p53 tumor suppressor gene. This is a very aggressive model, and you can see that the mice die on average of around 50 days after birth with a fulminant pancreatic cancer. So we established a preclinical model to test whether neutralizing LIF with a neutralizing antibody called D25 affected the progression of the disease. We could show that treatment of KPC mice reduced phosphostat-3 staining in the tumor, and you can't really see this here, but if you look here, you can see there are nests of tumor cells with no nuclear phosphostat-3 staining, but there are still some cells that are phosphostat-3 positive. So we set up a preclinical trial in which we used a combination of gemcitabine, a nucleoside analog that is currently used in pancreatic cancer therapy, and the anti-LIF monoclonal antibody at 25 mg per kg, administered 2 or 3 times a week. And we found that a combination of gemcitabine and anti-LIF antibody gave you a significant increase in survival of these mice. So that was certainly encouraging that LIF is in fact contributing to tumor progression. We also found if we pre-treated the mice with a combination of triple chemo combination that's currently used in human therapy, nab-paclitaxel, a microtubule poison, cisplatin, and gemcitabine, followed by anti-LIF antibody, we now get a significant increase in survival. And this is really the way it might be used in the clinic. We could show that LIF depletion led to a reduction in the number of cells in the tumor with stem cell-like properties. For instance, here is reimplanting cells, purified tumor cells, back into the flank, and you can see if you pre-treat the mice with anti-LIF antibody, very few cells can now form a new tumor. We also found that anti-LIF antibody treatment resulted in a significant increase in the fraction of the tumor tissue that was differentiated according to 3 independent pathologists. We also used a genetic model to eliminate expression of the LIF receptor in the epithelial cells, i.e. in the tumor cells. In situ hybridization using RNA-scope showed that LIF itself is expressed primarily in the stromal cells surrounding the tumor cells, although there are positive tumor cells, whereas the LIF receptor in red here is expressed exclusively in the tumor cells. And what we found was that when we conditionally knocked out the LIF receptor in the pancreas, there was also a significant increase in survival, particularly in the presence of gemcitabine, suggesting that the LIF target is the LIF receptor in the tumor cells. So we could also show, now looking at a time course of progression, that LIF levels increase steadily in the KPC mice over time, and that we can detect LIF protein in the serum of these KPC mice, suggesting it might be used as a biomarker. Of course, all of this was done in mice, so what about people? If we look at tumor tissue using a LIF ELISA assay, we can see there are elevated levels of LIF in the tumor samples, much higher levels than IL-6, it turns out. And we can also detect high LIF levels in the serum of pancreatic tumor patients. If we analyze those patients in a little more detail, we can see that the levels of LIF correlate with the disease status, and there is a trend towards a correlation with longer survival with patients who have low LIF levels. So to summarize this section then, we've shown that a neutralizing anti-LIF antibody prolongs survival in a mouse model. LIF levels are correlated with disease status. And obviously, the next step would be to develop a humanized anti-LIF antibody to test in clinical trials. And Northern Biologics, with whom I have no financial interest, in Toronto has developed such a humanized antibody, and it began trials in August in highly refractory cancers, including pancreatic cancer. So hopefully in the next few months, we'll see whether the LIF antibody does have any palliative effect on pancreatic cancer. So in the last few minutes then, let me turn to a second topic. So 40 years ago, this is the reaction that I discovered. Tyrosine is phosphorylated on the phenolic hydroxyl group, generating a phosphate ester, a very heat-stable and acid-stable linkage. But it turns out that in addition to tyrosine, not only serine and threonine, but also 6 other amino acids can be phosphorylated. Most people don't even realize this. They include the 3 basic amino acids, cysteine, glutamate, and aspartate. Very little is known about these. But histidine sparked our interest because it is a ring-like structure, very like tyrosine, and it was already known that it could be phosphorylated. Interestingly, histidine can be phosphorylated either on the N1 to form a phosphoramidate, a phosphorus-nitrogen linkage, or the 3 position. So you have 2 isoforms of phosphohistidine. Both of these are chemically very unstable at any pH below 7 or when you heat the samples. Histidine phosphorylation is best understood in bacterial systems where the 2-component signaling pathways use histidine phosphorylation to transmit a signal across the plasma membrane. And the surface receptor autophosphorylates on a histidine, and this phosphate is then transferred to an aspartate to generate the signal. But phosphohistidine is also known in mammalian cells. There are metabolic enzymes such as phosphoglycerate mutase and ATP citrate lyase that use a phosphohistidine intermediate. But there are also proteins that are simply modified on histidine, such as histone H4. The only enzymes we know that can phosphorylate histidine are NME1 and NME2, also known as NDPK and B. These are actually nucleoside diphosphate kinases. They're housekeeping enzymes which rephosphorylate nucleoside diphosphates like GDP with ATP. But these enzymes use a 1-phosphohistidine intermediate, and that phosphate can also be transferred onto proteins, onto histidine residues. We've learned that histidine phosphorylation in mammalian cells regulates many different sorts of processes in different parts of the cell, including surface proteins, ion channels, at the mitochondria, and also in the nucleus where histone H4 is phosphorylated. To summarize what we know, in the bacteria, there are these 2-component systems that carry out a phosphohistidine phosphoaspartate relay. In mammalian cells, we have histidine kinases, NME1 and NME2, that use the enzyme intermediate to phosphorylate proteins. There are at least 3 phosphohistidine phosphatases that can remove the phosphate, making this a reversible phosphorylation process, just like tyrosine phosphorylation. In a few cases, we know what the phosphorylation on histidine does. In this case, phosphorylation of this histidine in the tail of a potassium channel increases channel opening. So in the case of tyrosine phosphorylation, anti-phosphotyrosine antibodies have been incredibly useful in studying this process. And we were attracted by the idea that because phosphohistidine is also a ring structure, we might be able to make antibodies to it for a similar purpose. We had tried this 25 years ago, but failed, probably because the phosphohistidine we used was too chemically unstable. And so we needed a stable analog, and Tom Muir's group developed these phosphoryl triazolyl alanine analogs where the phosphorus is linked to a 5-member ring like histidine via a phosphorus carbon bond that's totally stable. And there are 2 analogs, 1-PTZA and 3-PTZA. And we incorporated these analogs into a random alloglide peptide backbone, coupled this to KLH to immunize rabbits. And much to our delight, we obtained antibodies that were specific for the isoform. So here is NME1. It's a 1-p-His intermediate detected by the 1-p-His antibodies. Phosphoglycerate mutase is a 3-p-His intermediate, not detected, and vice versa. The 3-p-His antibodies only detect phosphoglycerate mutase. And when we heat the samples prior to running on the gel, these are all blots, by the way, the signal goes away. So we use these antibodies to generate, or use the rabbits to generate monoclonal antibodies, both to the 1-p-His and 3-p-His forms. And here are a series of pancreatic cancer cell lines, in each case blotted with an unheated sample and a heated control. The major bands are NME1 and NME2, the enzyme intermediates themselves, but there are other bands that go away with heating. And in the case of the 3-p-His antibodies, both E. coli and 293 cells show a lot of bands that disappear with heating. So we think 3-p-His may be the major type of histidine phosphorylation in cells. So we now have 3 unique 1-p-His monoclonal antibodies and 4 unique 3-p-His monoclonal antibodies that are available. We've used these antibodies for immunostaining. So here are staining of HeLa cells with a 3-p-His antibody. You can see nuclear green staining, sort of a dotted pattern. And these 2 dots here turn out to be the centrosomes, which, like the spindle poles, stain positive for phosphohistidine, suggesting that histidine phosphorylation might be important in progression through the cell cycle and mitosis. We've used the antibodies to isolate phosphohistidine proteins from cells to see what the global phosphohistidine proteome looks like. And we're able to isolate around 800 proteins that contain phosphohistidine. Many of these proteins are involved in RNA metabolism, a significant number in the cell cycle, as we might have expected. And we found many proteins we knew were reported to contain phosphohistidine. So additional work that I won't show you has indicated that, or maybe it's here, yes, there are going to be a lot of additional phosphohistidine-containing proteins, and our estimate, and Claire Ayer's estimate, is that about 25% of all phosphate linked to protein in a mammalian cell, a HeLa cell, for instance, is linked to histidine, lysine, or arginine. And so there's a very large hidden phosphoproteome that we know almost nothing about that's probably going to be very important. So the obvious question is, what does histidine phosphorylation do? Is it used for short-term responses because it's chemically unstable? Are there phosphohistidine-specific binding domains like the SH2 domains for phosphotyrosine? Is it simply working through a very large charge change, +1 to -2? Or can it regulate divalent methyl ion binding, as we have shown in the case of that potassium channel? So I'll just finish then with one sort of more translational discovery we've made in collaboration with Mike Hall and his postdoc Sravan Thindupur in a cancer model. So Mike Hall has generated a mouse in which mTORC1 is activated by TSC1 conditional knockout and mTORC2 by conditional PTEN knockout. And these mice develop a very rapid hepatocellular carcinoma. And they investigated by proteomic analysis which kinases and phosphatases had altered expression in the tumors. And they found that the most highly upregulated kinases were the NME1 and NME2 proteins, and the most highly downregulated phosphatase was LHPP, a phospholysine phosphohistidine phosphatase. So we collaborated with them to show that tumors from these mice have elevated levels of 3-phosphohistidine as well as elevated levels of phospho-NME1 and NME2. It's a 2- to 3-fold increase here. To test whether LHPP was really responsible for dephosphorylating histidine in cells, it was expressed in a cell line from one of these tumors via an adenovirus vector, and this significantly decreases the level of phosphohistidine. And in vivo, re-expression of LHPP via injection of an AAV vector into the tail vein, which takes it to the liver, caused a dramatic decrease in the number and size of tumors in these mice. Of course, again, that was mice. What about humans? Well, Mike has a collaboration with clinical oncologists in Basel, and in these two patients, you can see that their tumor tissue has very high levels of 3-phosphohistidine compared to normal surrounding liver tissue. So what we would conclude is then that histidine phosphorylation is playing a driver role in liver cancer, and that the LHPP phosphatase is a tumor suppressor, which is downregulated in these hepatocellular carcinomas. So that leads to the question of how histidine phosphorylation is working and whether one might be able to develop drugs that antagonize this increased histidine phosphorylation. So let me finish then by just reminding particularly the students in the audience, how long progress in science can take. So the discovery of CML that Brian will talk about was back in 1845, and it took till 2001 through a series of discoveries like the Sark tyrosine kinase, the Abel tyrosine kinase, to develop a drug that antagonizes BCR-ABL and acts as sort of a miracle drug for CML. Let me then thank the people who did the work in the lab. Targeting LIF from stellate cells in pancreatic cancer was spearheaded by Yu Shi in the lab, with a collaboration from Rujun Tian, who was a postdoc in Tony Forson's lab and is now running a lab at SUSTech in Shenzhen. And the work on the phosphohistidine antibodies really was driven by Steve Foos from help from other people in the lab. Finally, let me thank the old buffer and the work on phosphotyrosine with my colleagues at the Salk, Walter Eckhart, Bart Sefton, and Marianne Hutchinson. Thank you very much.
Thank you very much, Dr. Hunter, for your marvelous lecture on tyrosine phosphorylation and also the exciting new work on histidine phosphorylation. Now, I'll give a brief introduction for our next laureate, Dr. Brian Drucker. Dr. Drucker received his bachelor and MD degree from UC San Diego, and then he went to Boston until 1993 when he joined Oregon Health Science University as a staff physician with a joint appointment in the Department of Cellular and Development Biology. Now he is a professor of medicine and director of the Oregon Health Science University Cancer Institute. He has received many recognitions and awards. He is a member of National Academy of Sciences, National Academy of Medicine, American Association of Physicians, American Society for Clinical Investigation, and investigator for the Howard Hughes Medical Institute. He has received the Medal of Honor from American Cancer Society, the Lasker DeBakey Clinical Medical Research Award, the Meyerberg Prize, the AACR Richard and Hinda Rosenthal Foundation Award, and the Japan Prize. As already been mentioned, he has made outstanding contributions in the development and application of the TKI, Gleevec, which was the first successful example of tyrosine kinase targeted therapy by small molecule inhibitors. Let us welcome Dr. Drucker to give his lecture.
Thank you. It is an honor to be here and to be honored with the Tang Prize in 2018 with my colleague, Dr. Hunter and Dr. Mendelsohn. I'm going to take a slightly different approach than Dr. Hunter, not being quite the scientist that he is. But I also want to continue on the theme that he talked about, which is about the amount of time that it takes for a breakthrough. And so part of what I want to do is to take you through what I'll subtitle the anatomy of a breakthrough. And so really what I'm trying to share with you is, why is it that I'm here on this stage as opposed to somebody else? Or why couldn't this be you in 5 years or 10 years or 20 years? And so I'll start with the concept that Dr. Hunter talked about, which is that breakthroughs take time. And so to illustrate that for you, I want to take you back about a century. I'll take you back a century and have you go and visit with a doctor. And when you go to visit this doctor, you've been diagnosed with a common infection. And this is what your encounter is going to look like when you go to see that doctor.
A contagious disease, an infection just like any other. I've seen cases where it was transmitted by an inanimate object. You mustn't be disheartened. There are many as badly off as you. Many.
Will I get well?
You must come here twice weekly for sweat baths, observation, and medical supplies. The treatment consists largely in rubbing yourself with this ointment.
Tell me, doctor, will I get well?
Rub a different part of the skin every night of the week so that no part of the skin is rubbed more than once weekly.
There is a girl in Munich with whom I'm in love, who loves me. We had planned to be married as soon as I graduate. Tell me, doctor. Tell me the truth. Can we ever get married now?
I'm afraid marriage is out of the question, Hans. You may dress now.
Does anybody ever get cured?
Of course. There have been many cures. Many.
Yes, many cures. And the reality is, if you went to the doctor in the early 1900s with an infection, that was a lethal disease. And if you go back into the 1900s, the leading cause of death in Taiwan, in the United States, were infectious diseases. Pneumonia, tuberculosis, diarrheal diseases. Cancer isn't even in the top 5 on this list. But if you fast forward now a century, heart disease is the #1 killer in most countries. Cancer is close on its heels. In the United States, certainly will overtake heart disease within the next decade or two. But infectious diseases have been relegated to largely eradicated, treatable, or cured. So one could evaluate what was it over the course of the last century that allowed infections to be cured or eradicated. And it truly was a broad-based approach. Early in the 1900s, most countries began sewage and wastewater treatments. By the middle of the century, we had refrigerators in most homes. These were the public health measures that made our food and water supply safe. In the early 1900s, I had told you could target bacteria specifically with drugs called antibiotics. You would have sent a review like the one that was sent to Dr. Hunter in your paper saying that's ridiculous. But by the 1940s, 1950s, antibiotics came along and began to treat common infections. And in the 1950s, the polio vaccine, and now numerous vaccinations for polio, smallpox, mumps, measles, and many, many others. But if you cast these as broad categories, that would be prevention, specific treatments or targeted therapies, and modulating the immune system. So quite a broad-based approach over the course of the century to make infectious diseases treatable or eradicated. Dr. Hunter showed you this general timeline of the disease I worked on called chronic myeloid leukemia. And again, a lengthy history of breakthroughs and discoveries, beginning with the first clinical description of the disease in 1845, the identification of B cerebrals that cause a molecular abnormality, and then ultimately specific therapy for chronic myeloid leukemia. If you look at the clinical description, first described 1845, on the left by Virchow and on the right by Bennett. And so I've got that right. On the left by Bennett, on the right by Virchow. These two manuscripts were published in 1845. Now, it's said that Bennett published his description 6 weeks earlier than Virchow. Now, think back, 1845, there wasn't exactly a computer. There were not really even typewriters. These manuscripts were hand set and printed. And can you imagine even how the reviews were done without a reliable postal system? Nonetheless, there ensued a vigorous debate about who was really first. And ultimately, Virchow capitulated that Bennett had described his 6 weeks prior, so Bennett gets priority. So another lesson to learn is debates about who was first in science go back a long, long ways. Now, fast forward about 150 years, we now know that chronic myeloid leukemia is one of the 4 common types of leukemia, making up about 15 to 20% of all leukemias. The incidence is pretty flat worldwide, affecting about 1 to 2 people per 100,000 per year. The disease can affect any age group, but the average age of onset is about middle age, 50 to 60. And historically, the median survival was 3 to 5 years. So, second precept, when you think about a breakthrough, is a breakthrough requires knowledge. Let's go back and visit our doctor in his clinic and see what he has to say about that.
Gentlemen, it would seem that the germ of syphilis has been discovered.
Really?
No. By whom? By one Fritz Schauding. The German Medical Weekly sent me these proofs for approval. Listen. The spirocheted pallida is a protozoa. It is a fine, steeply convoluted filament with 6 to 14 turns. It is decidedly motile, with forward, turning, and bending movements.
Tell me, does that description put you in mind of anything else?
It is decidedly motile, with forward, turning, and bending movements.
Why, that's just like the trypanosome.
Right. If the germ has been discovered, there's cause to hope for a cure.
Yes, there is hope.
So there we have it. If the cause has been discovered, there's hope for a cure. So as I'm talking about chronic myeloid leukemia, you're free to think about whatever disease you happen to be working on and can follow that same logic. So we need to understand what causes CML. And we'll start in 1960 with Peter Knoll and David Hungerford, who, working in Philadelphia, identified an abnormal chromosome in the blood and bone marrow of patients with chronic myeloid leukemia. And in 1960, they published this landmark paper in Science. This is the entire paper, 3 paragraphs. So yes, that's right, quality and quantity do not equate. But as you often do in the last paragraph of your paper, you think about the broad implications of your work. And what they said was the findings suggest a causal relationship between the chromosome abnormality observed and chronic granulocytic or chronic myeloid leukemia. As most scientists are skeptical, the general community thought that they were absolutely wrong about that. It turns out they were absolutely right and way ahead of their time. In 1973, Janet Rowley, working at the University of Chicago, showed that it was thought to be a shortened chromosome 22, the so-called Philadelphia chromosome, actually came about because of a reciprocal translocation between the long arms of chromosome 9 and 22, creating a shortened chromosome 22 and an elongated chromosome 9. By the 1980s, when individuals were mapping oncogenes to chromosome locations, it became clear that what happened as a result of this chromosome translocation was that the Abel tyrosine kinase located on chromosome 9 was translocated in its entirety to chromosome 22, creating a fusion gene and protein called BCR-ABL. And I'd point out that today is 9/22, and here we have the 9/22 translocation, and today is World CML Day. The next precept to think about a breakthrough is that breakthroughs often occur when different fields of investigation converge. But it's also, you have to apply the right technology at the right time. As you heard from Dr. Hunter, when he was beginning his career, sequencing an individual gene was a Herculean task. By the time he described 500 different kinases, we could sequence an entire genome. But let me take you through this timeline, some of which Dr. Hunter has covered quite eloquently. So out of tumor virology came the oncogenes BSARC and ABL. I take you through chromosome banding and chromosome analysis, which led to the discovery of BCR-ABL. And of course, the entire field of protein phosphorylation with Dr. Hunter's seminal finding in 1979 of tyrosine kinases with the identification of VSARC and ABL. All of this led to the notion that someday you could produce a tyrosine kinase inhibitor. So the last precept I'll cover is that breakthroughs require seeing things differently. And oftentimes when people give lectures like this, they'll say, you've got to think outside the box. Well, what I'm going to tell you is sometimes you got to see the box and call it a box, because the answer may be right in front of you, staring you in the face. And I can now add yet another example from Dr. Hunter's work where he could have discarded that gel that showed phosphotyrosine, but he took a deeper look. And I'll give you several other examples where individuals looked at something right in front of them and discovered something remarkable. I'll start with Velcro. We all use Velcro in our daily lives, but the history of Velcro was this: Georges de Mistral, a Swiss engineer, went out for a hike in the Swiss Alps one day wearing a velvet vest. And he came back from his hike, and his vest was covered with burrs. Now, like most people who've gone on hikes with their clothes covered with burrs, you pick them off, your finger gets pricked, and you swear at the burr and you go on with your day. Well, he asked this important question. He asked, why do these burrs stick so tightly to my velvet vest? Happens that he had a microscope down in his basement. He went and looked and found that his velvet vest had loops and the burrs had hooks. And he said, I bet I could make a fastener out of that. So, like many before him, he was the first to ask the important question of why. I'll give you a medical example. Penicillin. Alexander Fleming. The story goes that Alexander Fleming went into the lab one day, looked at a plate of bacteria that got contaminated with yeast, and saw that the bacteria weren't growing very well around the yeast. And instead of throwing away those contaminated plates like many before and thousands after him would have done, he again asked the question, why are these bacterial growth inhibited? And he discovered penicillin. But what people don't realize, people think that was a chance finding. But the reality is Sir Alexander Fleming was a physician, and he had done training during World War I. And he grew frustrated by removing bullets from wounded soldiers only to have them die 2 or 3 weeks later of an infection. So he dedicated his career to finding compounds that could inhibit the growth of bacteria. 12 years later was that fateful day that he walked into the lab. So was that an accident? Was that a prepared mind? It really doesn't matter. He went and asked the important question, why? So I came to the field of CML in 1988 to 1990, and here's what was known. BCR-ABL, the product of the Philadelphia chromosome, present in virtually every single patient with CML. It was thought to be, at least in my mind, the causative molecular abnormality, although at the time it was debated. And we knew that BCR-ABL functioned as an activated kinase. And one of the early experiments I did in the lab was to knock out the kinase activity and showed that it no longer functioned as a transforming oncogene. So if you think about this schematically, tyrosine kinases transfer phosphate onto tyrosine residue to specific substrate proteins, and it's these substrate proteins that cause all the abnormal proliferation that you see in patients with CML. So in my mind, I thought if you could block binding of ATP to this specific tyrosine kinase, you could have an ideal therapeutic approach. So in 1993, when I moved to Oregon, my colleague at Ciba-Geigy, Nick Leiden, sent me a handful of compounds, including this one, STI-571, or Gleevec, which ultimately became Gleevec. And what our lab showed was this was the best compound at inhibiting the growth of CML cells, both in vitro and in vivo in animal models, as well as eliminating CML cells from patients with the disease. But the problem was there were lots and lots of reasons this drug never should have been developed. The first hurdle, which actually was even getting a drug company to work on this, is that somebody had predicted there was going to be 1,000 kinases in the kinome. It was Tony Hunter. And what drug companies thought was if you go back here, if all these kinases bind ATP, if you block one at the ATP binding site, you're going to block them all. So the view was there will never be able to get a specific kinase inhibitor. So the general view was you cannot make drugs against this target. Oncologists are incredibly skeptical. And the view was there has never been a single drug that works against cancer in its history. Why on earth would a single drug for CML work? And then the problem also was that some of the early knockouts of tyrosine kinases, PDGF receptor, EGF receptor, were embryonic lethal. And so the view was even if you had specificity, these were going to be incredibly toxic. And if you didn't have specificity, they were going to be horrendously toxic. But the biggest hurdle was they're never going to make any money. And so imagine you're a big drug company, and your marketers are looking at the market for patients with CML, 5,000 new cases a year in the United States, 5,000 in Europe, and probably a couple hundred in Taiwan. So you look at the market and you say it's going to cost us $1 billion to develop this drug. The drug is going to have a 1 in 10 chance of success. And if the market penetration is about 30% in the first year, we may make $100 million a year. So is there anyone who wants to invest $1 billion with a 1 in 10 chance of success with the ability to make $100 million a year? I'm probably not going to get many takers on that. And that was ultimately the calculations that were done and was the biggest hurdle. But the reality is there were lots of patients in my own clinic that needed this. And fortunately, I was able to convince the company to go to clinical trials. And this is one of the earliest patients we put on. And it's often said you only need one patient to tell you that you have an effective drug. And so this is the before and after picture. The numbers on the left are the patient's white blood count. 10,000 is a normal white blood count. And I used to treat this patient with an old drug called Hydrea, and his blood counts would rise. I'd raise his Hydrea dose. When his blood counts would lower, I'd lower his Hydrea dose, and his white count would bounce back up. Now, his wife, who's an accountant, loves numbers, would keep track of me. When her husband's white count would go up, she'd say, "Dr. Drucker, you're not doing very well at controlling my husband's blood counts, are you?" And I would say sheepishly, "Ma'am, I'm doing the best I can." I'd raise his dose, and I'd usually overshoot. In April of 1999, he was started on 300 milligrams a day of Gleevec, and by December of that year, his wife finally said, "Dr. Drucker, you're actually doing a pretty good job. I'm going to stop checking on you." Now, of course, the FDA doesn't like anecdotes. This was our 5-year now survival data, and you can see we have a 90% survival with imatinib or Gleevec at 5 years. So remember, previously survival had been 3 to 5 years. In 2001, this drug received the fastest FDA approval in the history of the FDA and appeared on the cover of Time magazine. So let me introduce you to one of my patients. Oh, I'm sorry, one more. In our 10-year follow-up, we published this manuscript in the New England Journal of Medicine. And in patients in this column called Major Molecular Response, which is a 3-log response, look at these remarkable data. 100% of patients with a major molecular response or a deep response will survive 10 years without dying from CML. Now, the reality is not everybody survived. They died of other causes. But no patients with this response have died of CML, a truly remarkable finding. So now let me introduce you to one of my patients.
Hello, everyone. My name is Katie, and I'm very excited to be here tonight. I'm going to start out by telling you all a little bit about myself. I'm 18 years old. I'm a nursing student at University of Portland. I graduated in the top 8% of my high school class. Thank you. I was part of the Royal Crowns dance team where I was team captain, all-state athlete, and state champion. I love my dogs, Italian food, and Grey's Anatomy. And when I was 6 years old, I was diagnosed with chronic myeloid leukemia. We all know what happens in the treatment for cancer: chemo and radiation, hair loss and nausea. But would you like to know a secret? I never had any of that. I had Gleevec. Because of Brian Drucker, my life is as I described it to you. Dr. Drucker began developing Gleevec the same year that I was born, and it was FDA approved just 1 month before I was diagnosed. And if you want to talk about timing, that's some of the best that I've ever heard. It's because of the Knight Cancer Institute that this timing is possible. It's because of the Knight Cancer Institute that Dr. Drucker was able to develop this groundbreaking new drug. It's because of Brian that I got to carry a first place trophy back to my dance team at state. And it's because of the Knight Cancer Institute that I'm going to school to become a nurse. Can you imagine a world where cancer is merely a memory? Brian Drucker, Phil Knight, and you together are making this a reality, and I personally thank you.
Katie, thank you. Katie is now living her dream and is a nurse at the OHSU hospitals and clinics, working in our pediatric wards. So the reality, though, is that imatinib isn't perfect. When we look at our 5-year risk of relapse, it runs about 17% of patients will have resistance. What we've learned from the work of many around the world is that the major cause of relapse, about 60%, are mutations in the BCR-ABL kinase itself, and about 40% are other signaling mechanisms. What that's led to is the development of 4 new drugs to target the kinase-resistant mutations. So we now have imatinib as the current standard of therapy, significantly prolonging survival. We have relapses mostly due to kinase domain mutations, and we have novel ABL inhibitors that show substantial activity in patients with these mutations. All 4 have been FDA approved and are now being used in newly diagnosed patients. And this disease has been converted to a manageable condition. And even more remarkably, some of the patients with the deepest responses have been able to discontinue therapy, and it seems to be permanent, as it's been well in excess of 5 years off therapy, although that is a minority. So where else has imatinib worked? The 3 targets of imatinib are ABL, KIT, and the PDGF receptor. And we see activity in diseases driven by each of these 3 oncogenes. Gastrointestinal stromal tumors driven by KIT mutations discovered actually in Japan. Melanoma, a rare 2% have KIT mutations. And then a couple of skin disorders, hypereosinophilic syndrome driven by PDGF receptor mutations, and DFSP driven by PDGF ligand overexpression. Imatinib, though, has been tried in numerous other malignancies where it has not worked despite expression of the target, and that just tells us that the target expression is not enough. We need dependence of the cancer on the abnormal target. So what lessons have we learned? And for me, we have to identify the right targets. And if you have the right target and a good drug, you can get good results. It really can be that simple. But for as long as I've given these talks, you can tell I've put a lot of emphasis on the target. But there have been several recent examples where relatively poor drugs have almost killed good targets. So it's clear to me that we do need the combination of good targets and good drugs to get good results. So how do we translate this to other cancers? Well, first of all, we need the right targets. And to my way of thinking, these are going to be the early molecular pathogenic targets in cancer. But a lesson that often gets forgotten in all of this is that chronic myeloid leukemia is a model of an early cancer. It progresses over time from a chronic leukemia to an acute leukemia. We treated the chronic stage that has a 3 to 5 year lead before it becomes a full-blown acute cancer or acute leukemia. So to extend this paradigm and see the success that we've seen with imatinib, we have to treat earlier in the course of the disease as opposed to advanced metastatic cancer, where we see far more molecular heterogeneity. And to do that, we have to do much, much better identifying cancer at these earlier, more treatable stages. And of course, we have to do much better at matching the right patient with the right drug. So when you think about where we are, we're still relatively early in this evolution toward what people are calling precision oncology. We still do largely treat cancers by their site of origin. The treatments are still largely empiric, including many of our immunotherapeutics now, which are remarkable tools, but are still used largely empirically. And our response rates are relatively low, and we can't yet predict which patients respond. So as I think about the future, we have to think about taking a patient's sample and characterizing the tumor extensively. Not only the tumor, but the surrounding microenvironment, as well as surveying the immune status of the patient and the tumor. We need to turn that around as quickly as we possibly can, possibly even within 2 days, to make treatment decisions. So one of the things that we need to think about is taking that patient's sample, looking at the genome, looking at what's expressed, looking at what pathways are activated, looking at what the immunophenotype is of the cancer and the surrounding microenvironment, thinking about PDX models, thinking about screening, then running that through our computational algorithms and using that to design treatments. Now, you might think this is a little bit of science fiction, but in the next month, our group is publishing a paper in Nature where we've been looking at patients with AML. And we've been doing pretty much a fair bit of this. We've been taking the mutation, expression profiling, and functional screening and looking at what drugs patients might respond to. So that was 600 patients with leukemia. All that data will be made publicly available once the manuscript is published. So again, we're already beginning to see that accomplishing this is actually doable, and we are now turning that into a clinical trial in patients with AML, which is the most lethal leukemia, with a survival rate, if you're over 60, of about 10% at 5 years. So coming back to what we did in the last century with infectious diseases, where we took a broad-based approach, it's my view that in this century will be the century where we can do the same thing for cancer. We can think about specific targeted therapies directed at critical targets like imatinib and many, many others. We also have to think about modulating the immune system. As I mentioned, we've seen remarkable activity with some of the checkpoint inhibitors and other immune therapies, and we need to learn better how they're working and how to improve their ability to work. But we can't also forget prevention and early diagnosis. The best way to treat cancer is not to get it in the first place. The second best way would be to treat it early, but we also need to have that safety net for patients who are diagnosed with an advanced cancer. So in closing, I'd like to thank particularly all of my patients who went on this journey with me. All these are some of my phase 1 patients from 20 years ago now, who are still alive and doing many of the things they enjoy. This person gardening, this patient, the first patient from Italy who was treated with imatinib, who enjoys dancing. This woman from Southwest Washington who was referred to me from hospice. And these are her children from a picture in 2000. She now has great-grandchildren from the daughter on the right. Lots of patients traveling to Oregon to receive imatinib on our clinical trials. And some patients doing some remarkably and extraordinary things. This is the first patient treated from Australia in the year 2000. She returned to her home company and selected as one of the torchbearers in the 2000 Sydney Olympics. But the reality is I've shown you individuals, and when you start to think about this, it all begins to add up to groups. And for me, this is my hope for cancer. We'll take each cancer, each subtype of cancer, one at a time. But pretty soon it starts to add up. And we'll see far more pictures like this of patients surviving and thriving despite a diagnosis of cancer. Thank you very much.
Okay, great. Thank you very much, Dr. Drucker, for your most inspiring lecture, showing us how to pursue problems even when everybody says it cannot be done, and you succeeded. You made a marvelous breakthrough based on your ability to dissect the problem and pick the right approach. Now we have the third lecture, the Laureate lecture. The third laureate is Dr. John Mendelsohn. He received his medical training at Harvard University Medical School, and then he went on to receive medical training at Peter Van Brigham Hospital and Washington University at St. Louis, and then he became a professor at UC San Diego, where he was the founding director of the Cancer Center from 1976 to 85. After serving as a faculty at Cornell Medical College, he went to Houston to become the professor of genomic medicine and experimental therapeutics of the Division Cancer Medicine of the UT MD Anderson Cancer Center. He became the president of the UT Medical Center, UT Medical MD Anderson Cancer Center president in 1996, which he served until 2001. He has received many awards and honors, including the Royal Netherlands Academy of Arts and Sciences, the Medal of Honor from American Cancer Society, the AACR Prize for Translational Cancer Research, the Fulbright Lifetime Achievement Medal, and the Gold Medal of Paris, among many others. He's also an honorary, has an honorary doctoral degree from the China Medical University here in Taiwan. He is not able to give his lecture here due to health reasons, but we're very pleased and honored to have Professor Ming-Chi Hong to give the lecture. Professor Hong received his undergraduate training at National Taiwan University and then his graduate training from Brandeis University in biochemistry. He received additional training in MIT, and then he became a professor and chair of the Department of Molecular and Cellular Oncology at the University of Texas MD Anderson Cancer Center in Houston. He was elected as a member of Taiwan's Academia Sinica, and he is also an honorary director of the Center of Molecular Medicine of the China Medical University Hospital. Today he's going to speak to us on precision cancer medicine, achievements and perspectives. Let's welcome Professor Hong.
Is it right? Okay. So I'm extremely honored to be here today to present this Tang Prize 2018 Laureate lecture for Dr. John Mendelsohn. He's the President Emeritus of University of Texas MD Anderson Cancer Center. As Dr. Chen earlier mentioned, that because of his illness, he couldn't come today. So I'm extremely honored to be here to present his achievement, and I'll give a brief background introduction for himself, and also at the end, try to share with you his vision for the future cancer therapy. And I'm also pleased to mention that although Dr. Mendelsohn couldn't come to receive this award, that his son, Jeff Mendelsohn, who is right here, he was, yesterday he was in the award ceremony to receive the award for Dr. Mendelsohn. Jeff, send my best regard to your father. Okay, so as I mentioned, since I'm presenting this one for Dr. Mendelsohn, so I would like to spend just a few minutes to briefly introduce his background, although Dr. Chen Xu also mentioned a little bit already. John Mendelsohn came to MD Anderson 1996, and he served as a president for 15 years, stepped down at a time when he was 75 years old, and stepped down for presidency in 2011. But he did not retire. He continued to serve, and until this year, he just retired last month. He was the one who actually transformed MD Anderson Cancer Center, and now in the United States, for most of the time, the ranking for cancer center, MD Anderson was lucky to rank, always ranking #1, and Dr. Mendelsohn, is the major key person who pushed that MD Anderson into the stage. He graduated from Harvard College, bachelor degree, and then he received a very prestigious award, Fulbright Scholar, went to Scotland to learn molecular biology. At that time, it was an infant stage of molecular biology. Then he came back to the US, received the MD degree 1963. After his graduate from his MD degree, through his residence, fellow training, event at the end, Then he went to University of California, San Diego, to become the founding director of the Cancer Center. So he stayed there for almost 10 years, from 1976 to 85, and as a founding director for Cancer Center. Now, this cancer center has become one of the premier cancer centers in the United States. As a matter of fact, I will quote that today, one of our ORD, Dr. Brian Drucker, was a student there, and when Dr. Mendelsohn was a cancer center. Also, Dr. Tony Hunter, although he's not a member of Cancer Center, but he was a joint faculty with the UC San Diego. As a matter of fact, today, seems we have a lot of relationship with UC San Diego because our host, Dr. Chen Xu, was a professor from UC San Diego too. What a coincidence. Just like today was September 22nd, 922, Philadelphia chromosome translocation 922, where it causes the CML disease. Very nice coincidence. Then after 10 years served in the UC San Diego Cancer Center, he was recruited to the Big Apple. New York City, at that time, the #1 cancer center, always, that is Memorial Sloan Kettering Cancer Center in New York City. He stayed there as a department chair for medicine to run major medicine clinical trial and the major clinical activity. During that period of time, about 10 years, no question that Memorial Sloan Kettering was the #1 cancer center in the United States. Then, 1996, that year, I have served in MD Anderson for 10 years, and we are looking for our new leadership. 1996, MD Anderson is so lucky to be able to attract Dr. John Mendelsohn to move from New York City, go to the south, Houston, Texas, to leading MD Anderson Cancer Center as the president. Then he served, as I mentioned earlier, 15 years as a presidentship. And during his presidentship in MD Anderson, he actually transformed MD Anderson. Our size was increased almost 4 times during that 15 years. And he also moved the #1 title, quote, from Sloan Kettering, New York City, to Houston, Texas. Ever since now, MD Anderson in the United States, most of the time, we are fortunately ranking at the #1 cancer center. And probably a lot relate to Dr. John Mendelsohn. However, it is not really important to say who is ranking #1 or #2. What is the most important thing as a cancer center is how we can develop new therapy to treat cancer patient. I just give you one data, for example. 1 out of 3 drugs in the United States been approved by US FDA. The clinical trial has been done in MD Anderson Cancer Center. MD Anderson, today, we have almost 1,000 new clinical trials ongoing every year. And after he stepped down from presidentship when he was 75 years old, in 2011, he did not retire. Because his vision is to establish a personal cancer therapy institute. So then, after he stepped down from presidentship, he served as a director of personal cancer institute inside MD Anderson. Dr. Chen Xu has earlier mentioned his credit, so I just quickly say that some of his scientific achievement, that in early 1980, he and his colleague, Golden Settle, have made this first hypothesis that inhibition of EGF epidermal growth factor receptor that and of a tyrosine kinase could be an effective anti-cancer treatment. At the time when he made this hypothesis, it was in the early 1980, and then he went ahead, developed monoclonal antibody against EGF receptor, and then to show in the laboratory that has tumor suppression activity, and then went ahead to move into clinical trial. And as Dr. Brian Drucker earlier mentioned, even the Gleevec was an excellent drug which cures so many CML patients, but at the early stage that people are skeptical that how can a tyrosine kinase inhibitor that as an anti-cancer drug because it's going to be toxic and so on. The same thing applies to this monoclonal antibody, who at the time when Dr. Mendelsohn and Dr. Golden settled had this hypothesis. Actually, Dr. Mendelsohn told me at that time he couldn't get a grant if when you use the EGF receptor monoclonal antibody as a grant proposal because the reviewer are always very critical. This is impossible to use a monoclonal antibody to target a receptor as an anti-cancer drug. But now we all know monoclonal antibody is a standard methodology to develop drug, not only cancer, but on multiple different diseases, as long as you can against self-service receptor. And EGF receptor obviously is one of the prototype receptor that monoclonal antibody has been successful to be developed. And Dr. Mendelsohn was the key person, proposed the hypothesis, persistently develop antibody, move into clinical trial, and go to more than 2 decades to make sure this drug was approved by FDA to treat cancer patients. So C225 was at the time in the laboratory, they named this monoclonal antibody. In the lab, we sometimes still use this term, although now it's been humanized now. The FDA-approved drug is named as cetuximab or Erbitux. The first FDA approval was 2004 to treat colorectal cancer. 2 years later, head and neck cancer was also approved to treat head and neck cancer patient. And I had to mention the monoclonal antibody was developed 2 decades before the drug was approved. And because this is one of the prototype monoclonal antibody being as a drug. And so a lot of knowledge in the scientific community you need to learn how to make any antibody become a drug. For example, you have to humanize it and so on. Nowadays, monoclonal antibody is a standard method to develop whole kinds of medicine, and you don't need 2 decades to be approved. In addition to EGF receptor monoclonal antibody, Dr. Mendelsohn, while he was in Sloan Kettering, chairing the Department of Medicine, he has also actively involved the clinical trial of the Herceptin or trastuzumab. This is a drug developed by Genentech in San Francisco, target another receptor, tyrosine kinase called HER2 or NEW in breast cancer. HER2 is a brother of EGF receptor. The abbreviation HER2 is actually named human EGF receptor number 2. So you can see they're brother. They are the same family. And it was last new because at the time, almost at the same time that when the HER2 gene and red new oncogene was identified, was cloned, and the new was cloned by Dr. Robert Weinberg's laboratory in MIT. While I was a postdoc fellow, I was very lucky to be involved in cloning of this red new oncogene. So ever since then, I have been working on breast cancer. So Dr. Mendelsohn, in addition to EGF receptor, proposed the concept and all the way to push this monoclonal antibody become a FDA-approved drug. He also involved in other related receptor tyrosine kinase clinical trial. Dr. Chen Shi has earlier mentioned quite a few of his honor, let me just quickly mention a few things. That he was an American Cancer Society professor, and he received gold medal of Paris, and he is also an elected member of Institute of Medicine of the National Academy of Science in US, and now the name is changed into member of the National Academy of Medicine now. He was also an elected member Royal Netherlands Academy of Art and Science. He received then David Prize in cancer therapy in 2006, and also it's been earlier mentioned that in 2005, Dr. Mendelsohn and his lovely wife, and Mendelsohn actually visited Taiwan and received an honorable doctor and professor degree in China Medical University in Taichung. That year, he actually visited Taiwan's President Palace, and at that time, President was Chen Shui-bian. So, as a background, before I mention his hypothesis, his hypothesis was primarily based on that this earlier work from Dr. Cohen, Stanley Cohen, who was the pioneer person, identified EGF as a critical growth factor, and later on that showed this growth factor had to interact with cell surface receptor protein called EGF receptor, and this interaction turned out to be a very critical interaction to allow a growth factor go to growth factor receptor to stimulate cell growth and so on. And then through many scientists' contribution, that EGF or another ligand called TGF-alpha was shown to be able to form so-called autocrine model, i.e. tumor cell has a receptor by itself, overexpress ligand, and so make this tumor cell grow abnormally. And then, of course, that following Dr. Tony Hunter's discovery of tyrosine kinase in Salk, then Stanley Cohen that have also discovered that the EGF receptor has a tyrosine kinase activity, and later on, EGF receptor was shown to be overexpressed in many different kinds of cancer, including brain tumor, colon cancer, and head and neck cancer. So with all this background, so in the early 1980s, Dr. Mendelsohn and Gordon Sattel then make this hypothesis, that is monoclonal antibody, which can bind into EGF receptor and therefore to block the ligand EGF, or here the alpha, to prevent from cell proliferation and by inhibiting activation of EGF receptor tyrosine kinase. It's just a hypothesis in the early 1980s when he served as a founding director in UC Cancer Center, UC San Diego Cancer Center. And this concept then, with his team, started to generate this monoclonal antibody, as I mentioned, back to the old day, it is named C225. And please look at this panel. Here is a simple Western blot. And remember back to the old day that in order to detect a tyrosine phosphorylation, that was before monoclonal antibody against phosphotyrosine are available. This would have to use a cell, had to be incubated with a high radioisotope P32. And so then you can detect phosphorylating P32, radioactive P32 in the protein. So you can see here, when a cell stimulated with ligand, panel 1, no ligand, panel 2, you add a ligand, you do see the phosphorylation increase. You don't know what kind of phosphorylation, but you do see phosphorylation increase. However, in the panel 3 and panel 4, then when you start to add this monoclonal antibody, C225, which against EGF receptor, to prevent the ligand EGF to bind into EGF receptor, you do see this phosphorylation was reduced. Then in order to further validate what kind of phosphorylation this is, then use this I think this data, this technology Dr. Tony Hunter earlier has shown using this 2D electrophoresis, that you can see that this phosphorylation, the two spot, is well known earlier at the time that it's a serine threonine phosphorylation. And then after the ligand stimulation in this panel 2, you do see this extra spot, and this one is tyrosine phosphorylation, which originally was identified, developed, and this method was developed by Dr. Tony Hunter. And then now, if you add this C225 monoclonal antibody to block EGF to bind into EGF receptor, now you still see serine threonine phosphorylation. However, this tyrosine phosphorylation, which is critical to stimulate cell growth, now disappear. So this is a very preliminary result in the laboratory on the bench. And with that, then continue to move ahead. And here I want to make a point to thanks to Dr. Brian Drucker, that nowadays, when people to detect this phosphotyrosine, you don't really have to use the P32 label now. I think many people in the lab, if you are working in the lab in this area, you know you have a monoclonal antibody called 4G10. Now 4G10 monoclonal antibody, you can just easily to detect this phosphotyrosine. And 4G10 monoclonal antibody against phosphotyrosine was the one who developed by Dr. Brian Drucker. And my lab uses that all the time too. Okay, and so with this in mind, then Dr. Mendelsohn, and together with his group, they moved into a very pilot animal experiment. He was taking advantage of, at that time, the new mice, ophthalmic mice are available. So you can see, then when you take a tumor cell with the EGF receptor overexpression, you inject into the new mice, tumor develop. However, when you treat with this C225 monoclonal antibody, you see the tumor development was significantly suppressed. This indicates the tumor suppression activity. It was early 1980. Frankly speaking, nowadays in the literature, if you go to any cancer journal, any therapeutic paper, it was very easy to see this kind of data. However, back to the early 1980s, this is a very impressive data. And then, of course, with that, then he moved into clinical trial. And for the almost 10-year time, that start from hypothesis and produce the reagent to target the tyrosine, receptor tyrosine kinase, move the first clinical trial, and then this clinical trial had to back and forth because when you're doing a clinical trial, you realize that there's some issue there. For example, there's a side effect and the antibody had to be humanized and so on. So back and forth for the clinical trial, and eventually, at the end, that to show this clinical trial work, and then in 2004, the first FDA approval was occurred to treat colon cancer. In addition to this monoclonal antibody as a drug per se, that his lab, together with his colleague, has also shown a very dramatic result. That is, this monoclonal antibody against EGF receptor not only suppress tumor, in some cases actually can overcome some tumor cells which are resistant to certain type of chemotherapy. There are multiple examples like that, but here I just show one example. That is, for example, this tumor injected the mice, you see the tumor development. But if you treat it with the chemotherapy, doxorubicin, this particular tumor is known to be resistant to doxorubicin. So doxorubicin doesn't kind of suppress a little bit, but doesn't really suppress tumor because this tumor was resistant to that. If you use the EGF receptor monoclonal antibody, you suppress a little bit, not really work. But when you use the combination, you see the tumor is gone. So EGF receptor also has the ability to convert those tumor cells who resist certain type of chemotherapy to become sensitive. After the drug being approved by FDA. Now, this is a famous that the Hanahan Weinberg, so-called this hallmark of cancer. And so they summarize this hallmark of cancer, including all different kind of activity that could contribute to the cancer. And EGL receptor inhibitor, this including cetuximab monoclonal antibody, developed by John Mendelsohn, and of course the EGL receptor small molecule tyrosine kinase inhibitor. And in this hallmark of cancer, I should point it out, in addition to EGL receptor, this VEGF blockage and the HGF-c-MET blockage, both of them are also tyrosine kinase inhibitor. And I'm sure some other area, tyrosine kinase is also involved. And here I should also point it out that Tang Prize, this is the third time, give this particular area and tyrosine kinase and the blockage of tyrosine kinase activity, and to be a turning point of target therapy. And I still remember the first time when the Tang Prize was studied was awarded to the immunocheckpoint therapy for anti-CTLA-4 and the anti-PD-1. This one was originally discovered by Jim Allison over in MD Anderson, and then Jim Allison shared with Dr. Hongzhou in the Tang Prize first of all, and later on I will come back to this one. So here just show you how important tyrosine kinase is. It is hallmark, at least 3 of them directly involve tyrosine kinase blockage, and others are likely to be involved. This is a list, I think the data come from last year. All these slides are from Dr. Mendelsohn. That this is a list to show that US FDA approved those molecular target oncology agent. So as you can see here, and here is the target, here is the agent, and what kind of molecule that is. Out of 20 drugs, 11, i.e. more than 50% or about 50%, target tyrosine kinase. So i.e., if Dr. Tony Hunter did not discover tyrosine kinase, 50% of these drugs disappear. 1,000, 10,000, or might be millions of patients actually will be affected. And among them, 5 out of 20 was actually target EGL receptor, including cetuximab or gefitinib, the tyrosine kinase small molecule inhibitor, and primarily because EGL receptor is a prototype of receptor tyrosine kinase that have been served as a target therapy. That's why there are so many drugs against that. And I should also point it out, the most amazing tyrosine kinase inhibitor, Gleevec, imatinib, which Brian just mentioned earlier. As a matter of fact, I had to say, this Gleevec, imatinib drug, at the time when they treat with the CML, as the data show, this originally is a disease no cure, but now the 5-year survival rate, it's up to 90%. Even some of the patients will develop resistance, and their second generation of drug are continuing. This is an amazing drug which turn the target therapy field start to become very active rolling. Now, with all this stuff, at the time when Dr. Mendelsohn retired, he actually summarized in this kind of concept. Therefore, he would try to establish a so-called personalized cancer therapy institute. That is, he said, we have identified most of the genetic abnormality that cause cancer. And let me just give you a little bit of. In early 2000, when the sequencing has been developed, the entire human genome, 3 billion base pair, been encoded. Now, those cancer patients, what kind of mutation those tumor cells has can be easily by sequencing. And nowadays, probably in a couple of days, and maybe less than $1,000 depending on which place you do the sequence, you'll be able to determine your DNA sequence, the tumor DNA sequence quickly. I still remember in the old days, if Tony and Brian, I'm sure all of us, we have done this DNA sequence. back to the old day when Maxim Gilbert and Sanger just developed sequencing in the mid-70s to determine 1,000 base pair, it would take a whole year. It would take a whole year, I'm not kidding, because that's part of my PhD thesis too. However, today, 3 billion base pair for one human genome can be done in a couple of days. That's technology developed so fast. And with those kind of technology developed, so that the Mendelssohn field, now since we have so much data, we know what kind of, when the patient comes to the hospital, what kind of mutation can be easy to detect. And in addition to those FDA-approved drugs, that there are almost 800 different kind of drugs in the pipeline. When I say in the pipeline, they are either in clinical trial or they are in the preclinical development. Those drugs are already available to target product of the different kind of gene. For example, once you know a particular gene mutation or a particular gene overexpression, then you can develop inhibitor on that. So he thought that this particular molecule could be used as a biomarker. For example, using Philadelphia chromosome, that what Brian Drucker mentioned earlier, that chromosome 922 translocation BCR-ABL could be a biomarker to identify what tumor could be treated with the Gleevec and so on. So using this, and now as I mentioned, now relatively pretty short time and a reasonable cost, one can determine the tumor DNA sequence. So based on this one, there are a lot of examples, and the BCR-ABL is one of the good examples. HER2 overexpression is the other one, EGFR receptor overexpression, and another one. All those are patients who has abnormality of the biomarker that one can develop inhibitor. So here just summarize some of those very successful one, and as mentioned, this has been mentioned quite a few times, that Gleevec, that for CML, this is probably one of the best example for tyrosine kinase to inhibitor to treat with the cancer patient. So with all these available biomarker to allow us to identify right patient to be treated right drug. So at the time when Dr. John Mendelssohn retired in 2011, he would establish a personalized cancer therapy institute inside MD Anderson. And the goal is following, and this is a huge project, and it's still moving, and I predict it's likely to have a huge impact on the cancer therapy in the future. That is to create infrastructure and platform which could be able to systematically to genetically analyze the patient's tumor tissue by sequencing. And then to support, after you identify the tumor mutation, you should be able to support clinical trial and then bring the right therapeutic drug to treat the right patient. Then later on you see this is going to be generate a huge number of data, so it is important those huge database can be available to help the clinician to make a decision to support the personal cancer therapy. And as Brian earlier mentioned, even CML, even the Gleevec is such a dramatic anti-cancer drug. Eventually, some of the patients still develop resistance. Many receptor tyrosine kinase inhibitor all develop resistance, and that one would have to understand their mechanism, and so that to develop method to block resistance. And very importantly, although it's not scientific directly related, that we would like to demonstrate this value of approach be such a way that can become a standard of practice cancer patient treatment and can be reimbursed. Otherwise, cancer treatment can be extremely costly. So here, for this Institute of Personalized Cancer Therapy in MD Anderson, we initiated the following, and that this was led by Dr. John Mendelssohn after he retired from presidentship. At the time, he was 75 years old. So we started first use a 50 gene set. These are the gene, so all expectation for out of 2,000 patients with all different kind of cancer coming, sequence all these 50 genes. And here is the percentage of the mutation of a different gene that contribute to different type of cancer. Then from there, estimation approximately almost 40% of those mutations are considered to be actionable. When I say actionable, meaning potentially after you identify mutation, they may have the drug to be treated, okay? So as I mentioned, out of 50 gene panel, almost 40% are actionable, and then at the end, only 11% of the patient was really moved in clinical trial because of different kind of combination. Later on, when this project was ongoing, this 50 gene panel was expanded to 400 gene panel, and that increased this actionable gene almost 50% of the patient. This includes all different kind of cancer. So almost 50% of the patient are now potentially actionable, and then total 24% are enrolled in clinical trial. And in the next slide, I'd like to quickly share with you all these different kind of molecule, all these different kind of gene, either mutation or implication or abnormality. And here, the corresponding drug by different kind of company. So here involve almost 80 different kind of alteration, either mutation, implication, and so on. And then involve 44 different drugs from different kind of company and almost 50 clinical trials. So those patients come in, it's not like you're breast cancer, lung cancer, prostate cancer. No, you're a cancer patient. You come in, you sequence it. Then based on the sequence, you group them at a different kind of a clinical trial. This experiment is still ongoing, and I'm sure this is going to generate a huge number of data. And in the future, likely, we're hoping that either in the future for this, at this moment, this kind of experiment are very expensive, but in the future may become very cheap, and then maybe in the future, this is the way that to treat a cancer patient, that is, cancer patient coming, sequence, and then group, individualized. And if one inhibitor fails, you could try another inhibitor because the mutation is already known. That's one possibility. Alternatively, after all these data have been collected, I will not be surprised there may be some group that one can come out with a certain type of cocktail, either 2 inhibitor or 3 inhibitor, and then all these patients may be able to group at Group A, Group B, Group C, and Group A to treat it with one type of cocktail. Group B to treat with another cocktail combination. So under that situation, this so-called personalized therapy will not be that personalized anymore. It's become a group now. So those will be in the future that we can, after the data are collected, and then so based on this big data analysis, then from the data analysis that one may be able to come up with a strategy to treat with a cancer patient in the future. So that would have to wait and see. Is that going to be in the future all personalized therapy or maybe become a group therapy? Of course, ideally, if you can identify a set of cocktail and all cancer are using the same drug, that'd be a dream. And whether that dream can become true, I don't know. We'll have to wait and see. So here is a list of the dream that Dr. Mendelsohn, at the time when he retired, that's in his list. As Brian mentioned earlier, cancer therapy is important, but to have early diagnosis and prevention are important. So he was dreaming that using the biomarker to have diagnosis and in addition to treatment. And then this biomarker not necessarily have to be a tumor. Ideally, it could be from body fluid, for example, from serum or from urine. And as I mentioned earlier, it is important to integrate and share all this big data, including all the clinical response, biomarker, immunological, and imaging data. And then in the future, that those data could be developed and then could help to guiding physicians' decision to support specific treatment and so on. Most importantly for therapy, that is evidence-based combination therapy. And I would have to say, today, we're talking about tyrosine kinase target therapy. And then earlier, Brian also mentioned about immunocheckpoint therapy, and Tang's prize 6 years ago actually gave in this area. That is immunocheckpoint therapy. I believe combination of between these two or maybe other will be a very important, effective combination therapy for the future cancer therapy. And here, I would like to share with you, this is a very famous, the first Chinese medicine book. And I do not expect Brian or Tony or Jeff will read this, but I'm sure many of the audience know Huang Di Neijin. This book was saying, this was published almost 1,000 years ago. When you treat a disease, it is important to have a 正气存内，邪不可干. What this means is when you deal with the disease, two important factors. First one, 去邪, eliminate evil. Target therapy. So you want to kill the cancer cell. The other one is 扶正. What does 扶正? Strengthen body resistance, strengthen your immune response, and this will be immunotherapy. And this was predicted in the first Chinese medicine book, and I tried to find out when this book was officially published. Actually, I don't know. I don't know if it's 1,000 or 2,000 years because nobody knows it, but it was hanging around. Okay, so, and then here, I'd like to share with you the effort inside MD Anderson that we are trying to using this kind of combination therapy for treatment. As I mentioned earlier, that monoclonal antibody, such as satuximab, has now been a standard way. For immunocheckpoint, they also use a monoclonal antibody against the so-called PD-1 or anti-CTLA-4. And I don't want to go to those details. For those who know it, you know what is that. So this is a clinical trial outcome for melanoma. Keep in mind, melanoma, it is also a disease very difficult to cure. It's a solid tumor. There's no effective drug. Not until now, this immunocheckpoint therapy come out. So here's clinical trial data. So therefore, every spot, it's one patient's life. It's not mice. This is the clinical trial result. And this disease is known, has no cure before this drug was developed. So you can see here, anti-CTLA-4, pretty significant 30% survival. This is the 2015 data. Now the new data come out. This is about 44 years now. The line is continuing. Anti-CTLA, anti-PD-1, it's about 50 to 60% combination. It's close to 50, 60%. What the data tell us is, a disease no cure. Now with this kind of treatment combination, that one can reach to 50 or even 60% survival for up to 4 to 5 years. Not as good as CML, leukemia, but for this type of solid tumor, this type of data at this moment in the field was considered probably the best clinical outcome for the solid tumor. And today we are celebrating the 3 laureate who developed tyrosine kinase inhibitor and create so-called target therapy. And that make cancer therapy one big step ahead. But earlier we had talked about when we had to look at an area, one had to think about out of box and should not forget in the box either. So as a scientist, I think we should just think of whatever you want to think. This is a very impressive data for a disease no cure, then your new drug help patient to survive for 60%. But why not 100%? What's wrong with that? Can we achieve that? Let me use today's story as an example. 40 years ago, Tony discovered phosphotyrosine, discovered tyrosine kinase. 40 years later, there are tons of anti-cancer drugs based on tyrosine kinase, and the best example is that CML Gleevec, and including satuximab developed by John Mendelsohn. Are we going to wait for another 40 years from this 50, 60% up to 100%? This is a tough question, but if you ask me, my answer is no. It is not 40 years. I don't know when. I'm not saying next year. I'm not saying tomorrow. But the time is coming. So here I just show you that some approach we're thinking that we know solid tumor are heterogeneity. So one way to do it, and earlier that the speaker has been, our laureate has been already mentioned about effective combination therapy. And this particular trial is one of the effective combination therapy. And here I want to show you a little bit effort in MD Anderson. And MD Anderson has, as I mentioned, we have more than 1,000 clinical trials and about close to 200 clinical trials on immune checkpoint. And then in preclinical study, we have shown that, that's very nice, don't worry about this one. Here I just want to show, this is one of the combination therapy between a kinase inhibitor plus an immunocheckpoint therapy. And to deal with the disease at this moment, also no cure. That is triple negative breast cancer. For those of people who know breast cancer is not a big disease now. However, triple negative is still no effective therapy. Then this survival curve, what I share with you, is a model, this is an animal model. You don't treat them, they'll die. But when you treat them with metformin, which is a diabetes drug, and it's already known metformin can activate AMPK. Then by mechanism-driven study, we've noticed that metformin activates AMPK, can downregulate PD-L1 and enhance CTL activity. So if you use metformin by itself, it doesn't work. But then when you use anti-CTLA-4, you see the 10% survival. And this survival is 1 year tumor-free. However, if you use a combination between metformin and anti-CTLA-4, this is 50% tumor-free. And this triple negative disease, and this disease at this moment has no effective drug. The same combination therapy can be applied to other cancer types, colon cancer and melanoma. So this is one example that target therapy and immunocheckpoint therapy combined together. Alternatively, I'll share with you another one that is what we say about combination therapy. Either they are two drugs added together as a cocktail, or make one drug, two drugs link into one drug, and so become a multiple function. In this particular case, this is the anti-PD-L1 antibody as a therapeutic agent. Now, you added a very strong chemotherapy drug, 4 molecules per one. So now this is so-called ADC, antibody-drug conjugate. For this particular drug, when we treat the animal, and just to save the time to show the survival curve here. Again, this is triple negative breast cancer model. You don't treat them, they'll die. You treat the antibody alone. This antibody has been tested in human. There are multiple, there are at least 5 drugs have been approved by FDA, this kind of anti-PD-1 or anti-PD-L1. And in breast cancer solid tumor model, it's about 20–30% response rate. So this animal data actually recapitulate the human data. Now, my friend, please look at this red line. This one is this drug, antibody plus and antibody drug conjugate. This is 80%. And this is a disease at this moment has no cure. A data like this in triple negative breast cancer, now we have extend to colon cancer and melanoma cancer model, results are similar. Here I also want to share with you that all this model is model. Model is not real. Because this model you're using at one single cell line with a PD-L1 positive. However, we do know solid tumors are heterogeneous. Not every tumor cell expresses PD-L1. Some tumor cells express, some don't. So if you use immunocheckpoint, you are primarily target PD-L1 positive tumor cell. But those PD-L1 negative tumor cells, you cannot kill them. Therefore, at the time when we develop this drug, we on purpose to keep in mind that that can induce a so-called bystander effect. Bystander effect, in a very simple term, is like this. Assuming this room is a solid tumor, And each person is one tumor cell. And the one who wear glasses is PD-L1 positive. The one without glasses is PD-L1 negative. So if you use an immunocheckpoint anti-PD-1 or PD-1 antibody, you should only target the one wearing glasses. The one wearing glasses will be killed. The one without glasses survive. However, bystander effect, meaning I don't give it, excuse my language, I don't care, just I target the one wearing glasses, but when the one wearing glasses, that PD-1 positive tumor cell be killed, those PD-L1 negative cell next to the PD-1 positive cell was also killed. That's called bystander effect. So simple term. So here, just quickly show the data, the bystander effect. So if I use this antibody drug conjugate to incubate it with the tumor cell in vitro, then they are PD-L1 positive, so those tumor cells are all killed. This is yellow color. However, this tumor cell, if you knock down PD-L1, so they no longer have PD-L1, otherwise it's the same cell. You use this drug, you cannot kill them now because there's no PD-L1. So you kill the PD-L1 positive with glasses, this one without glasses, you don't kill them. Now, you mix them one by one, put together next to each other. All of them are killed. It's a red color. 0409 culture, the same result. Here's the in vivo result. It's similar. So now we have this drug, which in the animal model reached 80 to 90% in solid tumor model, and this induced bystander effect. The antibody has been humanized, and we are looking forward to be able to move it in clinical trial. So today we get together to celebrate the 3 laureate who has pioneer work, who had transformed our cancer therapy to create so-called target therapy. And then I think that we should work together to be able to make a cancer history. So at the very end, I want to take this opportunity to congratulate Tony for your discovery of tyrosine kinase, and Brian, your unbelievable clever drug to cure a cancer patient up to 90%, and of course, Dr. John Mendelsohn for his development of the cetuximab monoclonal antibody and his leadership in the entire cancer area. At the end, I'll say congratulations, and to 3 of you, I show salute. Thank you.
Thank you very much, Dr. Hong, for your marvelous lecture on behalf of Dr. John Mendelsohn. Ladies and gentlemen, we had a wonderful session this morning with fantastic lectures given by Drs. Hunter, Drucker, and Hong. These lectures are truly inspiring and stimulating. It showed us how the tyrosine kinase system was developed and then applied for the treatment of cancers through the use of inhibitors and receptor blockers. And the end result is to improve human health by combating this dreadful disease of cancer. It gives us many inspirations, such as how to overcome difficulties, whether it's reviewed by journals or the drug developers said no, and all the other things. But if we have the right way, we should persist and work on it. So this is not only for those working on cancer, but on biomedical research in general. These are most inspiring. And the other thing is we see how outstanding basic science developed can be translated to clinical medicine to benefit patients. So we should all follow this kind of example. This is indeed marvelous examples for us to follow. So today, this lecture session is really extremely beneficial to all of us. It is to the researchers, eventually to the patients. So with this, we'd like to congratulate and thank our remarkable laureates, and they are fulfilling the spirit of the Tang Prize, that is to improve the welfare of humankind. That is, in this case, through biopharmaceutical activities, but all of these Tang prizes are aimed at improving the humankind in their health and well-being. So let us, ladies and gentlemen, please join me in expressing our appreciation, congratulations, and gratitude to the three laureates, Drs. Tony Hunter, Brian Drucker, John Mendelsohn, represented by his son Jeff, and also Ming Chi for giving the lecture. Let's give them the warmest thanks and appreciation.
Thank you very much to Professor Chen for hosting this lecture session for us. And thank you again to Dr. Hunter, Dr. Drucker, and Dr. Hong for your inspiring lectures. Ladies and gentlemen, this is the end of the Biopharmaceutical Science lecture. Thank you very much for your participation today. If you have any questions regarding the lecture, you're welcome to join the press conference on the first floor.